Bismillahirrahmanirrahim. Assalamu alaikum. My dear students, you all know me, Talat Naz, Department of Physiology, SMC JSMU. This is the lecture that I have already delivered in the class. I am recording this for your request, salivary secretions. I hope it will be helpful for you. So, let's get started. Learning objectives of this lecture. By the end of this lecture, students will be able to understand the types and secretions of salivary glands, composition of saliva, stages of saliva secretion, neural regulation, and flow and reflexes responsible for the secretion or increment in the secretion of saliva. Introduction. The important glandular secretions involved in the GIT proper functioning may include salivary secretions, gastric gland secretion, exocrine pancreatic secretion, and liver secretions. These are the main secretions that are involved in the proper digestion of the food products. But our today's target is the salivary gland and their secretions. And you know the salivary secretion is saliva. So this is a cute baby and the saliva is coming out of the mouth. So this is the best picture I find for the saliva. And the saliva is basically extracellular fluid, and it is produced and secreted by salivary glands in the mouth. Basically, all the salivary glands are not present in the mouth, but the saliva is secreted into the mouth with the help of ducts, salivary ducts. So, if the saliva is produced by external glands, means the salivary gland is present outside or inside. In both the cases, saliva will reach to the buccal cavity by the help of salivary ducts. First of all, we will have a look on the physical characteristics of the saliva, how the saliva look like. Saliva is basically a colorless liquid and it is moderately viscous. To understand how viscous it is, you can take the example of the corn soup. So the thickness of the saliva is almost less like the thickness of the corn soup. So the pH, as far as pH is concerned, initially the pH of the saliva is slightly acidic, which is you can say between 6 to 7, and the average value of the pH is 6.7. Right, in the you can say in the when the saliva is newly formed by the acinar cells, and we are in the state of resting, means we are not eating anything, we are not chewing anything. In that condition, initial stages pe the saliva is quite acidic, but its pH become basic during activity of the glands, means when the huge amount of saliva is produced and transported through the salivary ducts, it shows that the activity of the gland is increased, right? In that case, what happened, the pH of the saliva will become basic, or the value will be increased to 8. And the major factor to make the saliva basic is the bicarbonate, presence of the bicarbonate. We will discuss this point in our later on slides. And about 1 liter of saliva per day is produced in our mouth, and it can be increased up to 1500 ml, that is 1.5 liter saliva. You can see the bottle of the cold drink. Everyone see the cold drinks in the 1.5 liter bottle. So imagine the volume of the saliva each day produced in our mouth, and it is so helpful in several functions that we will discuss later on. But here, this the physical characteristics. Yes, it is hypotonic to the plasma. Hypotonic to the plasma means it contains less amount of sodium and chloride and more amount of potassium and bicarbonate when we compare its concentration with the plasma, that is the isotonic plasma. So the physical characteristics shows also that the saliva is hypotonic to the plasma. If we undergo the chemical characteristics, we observed that saliva is basically consisting of two factors, water and solid material. Water is about 97 to 99.5%, and the solid material is 0.5%. Solid material is basically the proteins, right? And the different ions, different chemical composition, all these are basically the solid part. So it is further divided into two types. The material which is responsible for participating in the chemical characteristics are divided into two parts. That is, the solid part is divided into inorganic and organic part. The inorganic part is basically consisting of the buffers. You know that the function of the buffer is to neutralize the acidity and the basicity, that is to make it moderate for the functioning, right? So the buffers are present in the saliva, which is bicarbonate. As I told you that initially the saliva is acidic, so due to the uh, secretion of bicarbonate into the ducts, salivary ducts, it become basic in nature, so it will neutralize by bicarbonate. And then we have soluble calcium salts present in the saliva, which can help protection of the teeth, because you know the teeth are basically have a huge quantity of calcium in it. You know, all the bones of the body have deposition of the calcium. So the power of the teeth is also uh, due to the presence of the calcium. So we have different calcium salts which can help in protection of the teeth. And then we have some electrolytes like sodium, chloride, bicarbonate, and potassium. All these are considered as all these ions, because you know the bicarbonate, bicarbonate, calcium phosphate, sodium chloride, all these are the ions. They are all under consideration of inorganic substances. And then we have some organic substances, which is mucin. Mucin is also a type of the protein. And then we have some digestive enzymes like ptyalin and linguine lipase. They both participate in the digestion, also present in the saliva. So we can say that a minimum amount of digestion is started in your mouth with the help of digestive enzymes present in the saliva. And then come to the antimicrobial agents in the present in the saliva are the lysozymes, lactoferrin, and IgA. So these are basically the antimicrobial agents. And then protein rich protein, basically proline rich protein is a substance which constitute about 70% of the salivary protein. Means all the proteins which is present in the saliva when we see the composition, we see observe that proline rich protein is constitute about 70% of the salivary proteins, and it play important role. Now come to, as we have already seen, the physical and chemical characteristics of the saliva. Now come to the point from where the saliva is produced and how it is transported up to the mouth or buccal cavity. So here this picture is showing you different glands and different ducts, and we will observe this in our next slide. Salivary glands. Salivary glands are basically responsible for the formation, or you can say production, of the saliva. Salivary gland is any cell or the organ discharging a secretion into the oral cavity. ठीक है? What does it mean? That sometimes a single cell is responsible, goblet cell, yes, sometimes a single cell is responsible to release some amount of secretion that participate in the functioning of the saliva, right? So we can consider that cell as well as the organ, complete gland that is secreting saliva. So the salivary gland is any cell or the organ discharging a secretion into the oral cavity. So, but of course, there are some major glands and some minor glands. The major glands are basically parotid gland. There are two pairs of parotid gland, one on each side of your mouth, and it is parotid is near to your ear. It is anterior to the ears, and then this is the largest. Means parotid look like very large, but the amount contributed in the saliva is less as compared to small glands. So other one is the mandibular. The main contribution in the secretion of saliva or the content of saliva or the amount of saliva is basically from the submandibular gland. Submandibular gland is basically located near to your submandibular teeth. And then we have sublingual, as the name indicate, lingual means tongue. So sublingual means below the tongue. So this is the sublingual gland, and you can see these are the ducts that are coming from the parotid gland, from the submandibular gland, right? And the small ducts are sublingual ducts are also releasing their content into the oral cavity. Now, what are these? Are the major glands, that is parotid, submandibular, and sublingual. The other glands, the minor glands, are sometimes those in the tongue. Means some cells in the tongue are also responsible to secrete some secretions that are participating in the salivary function. And then we have some Tylen tonsil. Okay. Sometimes palatine tonsil, and then palate, lips, and cheeks. What does it mean? You can say and also a buccal, you can say buccal glands. Small buccal glands are basically all these cells are considered as buccal glands, because there are different types of cells which are contributing in the salivary secretion, but it is almost 7%, not more than that. Salivary secretory unit. What is salivary secretory unit? Basically, salivary secretory unit is consisting of acini and duct. So you can say that there are two major factors that are responsible to produce saliva and transportation of the saliva up to the oral cavity. One is the acini, and another one is the duct. So acini is basically consisting of acinar cells, and it secretes primary secretion which is isotonic to the plasma, means very near to the concentration of the plasma. Means the concentration of sodium, chloride, potassium, bicarbonate, all these concentrations are almost similar to the concentration of plasma. In addition to this, acinar cells also secrete some enzymes, or you can say prepare some enzymes with the help of course, mitochondria, ribosomes, and other cell organelles that are responsible to produce proteins. Enzymes are also protein. So all the contribution you have already have a background how the protein is synthesized. So enzymes, mucoproteins, electrolytes, all are collectively secreted from the acinar cells, and these secretions are basically considered as primary secretions because their composition is quite similar, or their concentration is quite similar to the isotonic to the plasma. Then we have different types of ducts, but striated ducts basically are more contributing in regulating the primary acinar secretion. Means there is regulation of primary acinar secretion means that when the secretion is released from the SNR cells, ductal modification will be take place, because this SNR cells release their secretion into the duct. So there will be regulation of this primary SNR secretion with the help of striated ducts and other ducts also. So as I told you that there are some major salivary glands, some minor salivary glands. Now we will see the contribution of these glands in the formation of saliva. First of all, we must know that saliva composition is consisting of water and solid material. So here we can see that the parotid gland, which is contributing to the formation of saliva, is about 20%. And the type of saliva or the composition that is secreted is about serous type. Serous means watery secretion, and it lacks the mucin protein but contain enzymes and ions. Then come to the submandibular and buccal glands. Basically, buccal gland contribution is very less, almost out of 75, you can say 7% contribution is from the buccal glands. And then you can see the submandibular gland is basically secreting serous secretion as well as the viscous secretion. So it is mixed type of secretion, and it is moderately viscous saliva that contain mucin, enzymes and ions. So basically, the thickness of the saliva is under the control of the mucin. The If the mucin is present in the saliva, of course it becomes viscous. If its amount is less, then it is moderately viscous. However, in the sublingual case, we see that the saliva which is produced by sublingual gland is about 5% of the total plasma that is produced per day, and it is viscous. And as I told you, that viscous is basically due to the presence of mucin. So here, the major part of the sublingual saliva is mucin. However, the mandibular is both mucin and serous secretion, and in the parotid we have the major content from the parotid gland, which is serous in nature. So you have to remember which part contain mixed and serous, and the mucin is basically purely contributed by the sublingual gland. However, a small amount of mandibular gland also produce mucin, enzymes and ions. So all these contribute in the formation of final saliva that enter into our buccal cavity. I have already discussed this part. As in our cells, first of all secrete the primary secretion, and then there is modification in that primary secretion with the help of salivary ducts. And this is basically the active process that is required by required energy so that the work will be done. Here we can see the ions and the salivary flow. As the saliva passes through the salivary duct, this part is the basically salivary duct, and this part, initial part, is acinar cells, right? These are sac-like structure where the saliva is produced and it is stored here, and then it will move into the duct. So the saliva passes to the salivary duct. Cations, sodium and chloride, are reabsorbed into the adjacent blood vessel. As I told you already, that when this saliva is produced with the help of these cells, acinar cells, then it will move into the direction of the duct. So when it enter into the duct, these are duct cells. What happened? You can see the sodium chloride is moving into the cells, and then it will enter into the blood, from where it will enter into the blood. And then there is entry of potassium and bicarbonate into this saliva. So basically the amount which is primary isotonic fluid, when it moves towards the duct, it becomes hypotonic in nature. The final hypotonic solution will reach into the oral cavity. Almost you can say it is hypotonic, not very much hypotonic, but its composition is entirely changed as compared to when it is produced by the acinar cells. because as I told you that initially it is acidic in nature, and then when it due to the presence of the bicarbonate, it become basic. So I was planning this picture, but I have to stop. So here we see that once the primary secretion has been prepared by the acinar cells, this will enter into the duct, and the modification of the primary secretion is take place into the ducts. First of all, the reabsorption of sodium occur by the active process, and chloride by passive process, and it will create electronegativity, right? And to compensate the electronegativity due to the removal of sodium from the primary secretion is contributed uh, electronegativity, and to compensate this electronegativity, chloride ions are also moving out of the duct, so that it will create again the positivity. However, the other part of potassium secretion and bicarbonate secretion also take place. So potassium will enter actively, due to which there is passive as well as active movement of bicarbonate take place, and this bicarbonate will contribute to the balance of this negativity, and also the bicarbonate is also participating in as a buffer. So basically the whole story of today's lecture is around the you can say reabsorption of sodium and chloride from the ductal cells into towards the blood, and the secretion of potassium and bicarbonate from the blood into the ductal cells, or the duct you can say. And then once these are enter into the salivary duct, the saliva become hypotonic, which reaches into our buccal cavity. That how the concentration has been changed, like net result resting condition में क्या होगा, once the saliva is formed and we are in the resting condition, we are not eating anything, then what will happen? Sodium and chloride ion concentration reduces about 1/7 of that plasma, right? And it will reach up to only 15 milliequivalent per liter each day. However, in on the opposite side, we see the potassium ion concentration is increased 7 times of the plasma concentration, which become 30 milliequivalent per liter. And similarly, bicarbonate ion concentration increased 2 to 3 times that of the plasma. So here we see that chloride and sodium concentration is decreased as compared to plasma. However, potassium and bicarbonate ion concentration is increased too much as compared to the normal plasma concentration. So all these contribute and lead to the formation of hypotonic saliva. And then during maximum salivation, when we are eating something, what happened at that time? The rate of formation of primary secretion from acini increased 20 times. Means the saliva which is formed by the acini will be increased 20 times. Its amount is increased by 20 times. Okay, due to the rapid flow. So of course the large amount is produced and it is rapidly moving into the duct. Due to the rapid flow of primary secretion from the duct, ductal conditioning become reduced. Means the conditioning means the movement of sodium, chloride, reabsorption, and the secretion of the potassium and bicarbonate is reduced. So what will happen? The ductal conditioning is reduced. Hence the saliva sodium chloride concentration is reduced, but only 2/3, and the potassium concentration become increased but only 4 times. However, in the resting condition, we have observed that the potassium concentration is increased 7 times, and in the maximum salivation time, the potassium concentration is increased only 4 times. So this is how the change in the concentration occur from the saliva, from the acini till the buccal cavity. सो बेसिकली अनस्टिमुलेटेड फ्लो होता है और एक स्टिमुलेटेड फ्लो होता है. ड्यूरिंग द अनस्टिमुलेटेड फ्लो वी हैव नो स्टिमुलस, right? Still the saliva is produced and moving into the oral cavity. Sometimes hyposalivation occur, not producing enough saliva. The condition in which saliva is not producing enough, it is called hyposalivation. And in the resting condition, when there is no need of saliva or very little need just to speak, to swallow, to clean the mouth, in that condition only then the hyposalivation is occur normally. However, and when there is a stimulus like taste, chewing, medication, any tactile stimulus, putting anything in your mouth, all these become the stimulus and increases the formation as well as the release of saliva into the buccal cavity. Less than 0.7 ml per minute is considered hyposalivation. Now see the difference. Less than 0.1 ml per minute means the person has hyposalivation in the condition of resting, right? And 0.7 ml per minute is considered hyposalivation in the active state. So you have to compare these values. Now come to the neural regulation. Neural regulation is basically the powerful side is from parasympathetic pathway. Basically, the signals or the stimulus is like smell, taste, sight of the food, and tactile stimuli like you are putting some hard, crunchy or saltish or whatever food you are putting in your mouth is conveyed through its signals are conveyed through the facial and glossopharyngeal nerves as well as facial nerves. Sorry, I didn't put here facial nerves, but facial nerves are also contributing in I think submandibular, the main role in submandibular. So the facial nerves. Sorry. Actually, it is night time. So what happened? Facial nerves and glossopharyngeal nerves, as well as vagus nerve, is also contributing to the salivatory nuclei in the brainstem. So it leads to the discharge of the parasympathetic nerves, increasing the rate of the secretion by 20 folds. Parasympathetic stimulation dilate the blood vessels. So the point is that the main contributing neural regulation is from the parasympathetic, particularly facial and glossopharyngeal nerves. And you have to remember the salivatory nuclei are present in the brain stem. So the signals are basically coming from the salivatory nuclei of the brain stem via the facial and the glossopharyngeal nerve that will be responsible for the discharge for the release of the saliva into the oral cavity. So, due to the parasympathetic activity or the signaling, the 20 times more saliva will be released into the buccal cavity. One more thing that the parasympathetic stimulation will do is to dilate the blood vessels around the ducts, glandular ducts, so that the more amount of sodium and chloride will move out of the ducts, and the more amount of potassium and bicarbonate will be available for the proper functioning of the salivary glands. So that's why the blood vessels become dilated and more bicarbonate will be available according to the requirement of the food. Here we see this is just to memorize that the salivatory nuclei are basically located approximately at the juncture of the medulla and pons, and they are stimulated by both taste and tactile stimuli, okay, from the tongue and other areas, as I already told you all these things. So this is the display of how the stimulus is coming and causing the stimulation of the parasympathetic pathways. Taste and tactile stimuli from the tongue and other areas of the mouth and pharynx will excite the salivatory nucleus. Stimuli from esophagus and stomach due to stimulation of the vagal afferent fibers will also cause stimulation. Stimuli arising from the higher center of the brain due to the sight, smell, and thought of the food can also cause the excitation of the salivatory nuclei. So here basically in this slide we want to discuss the reflexes. Which reflexes serve as a stimuli for the salivatory nuclei to be excited? Though So here we can see that the taste and tactile stimuli from the tongue and other areas of the mouth and pharynx, right? This will be one of the factor that can cause the excitatory signals towards the superior inferior nuclei. And then we can have sometimes we have normal and abnormal signaling from esophagus and the stomach, and this is called unconditioned reflex because these are sometimes abnormal. You can say this is not a normal reflex that esophagus will pass certain signals to increase the saliva, but sometimes due to some irritation, vomiting, some disease condition, and sometimes we have eaten which is wrong food, irritating material. In that condition it will occur. And sometimes we have food in front of us, and already the brain see the beauty of the food and the smell of the food, and sometimes we are just thinking that we are going to have a dinner. In that case also the brain will start to give signals to the salivatory nuclei, and it will become excited and release more and more saliva. The other part is the sympathetic. Of course, there is a little bit contribution of sympathetic pathway also in the neural regulation of salivation. Slightly increased salivation will be take place, slightly, very slightly, by the activity of the sympathetic pathway. These nerves, sympathetic nerves, are basically originating from the superior cervical ganglia, travel towards the blood vessel supply to the salivatory glands. So basically, superior cervical ganglia say nerves are originating, and they are supplying to the blood vessels. So basically, the role of sympathetic pathway we observed is that it causes the constriction of the blood vessels, and sometimes it can even stop the salivation or inhibit the salivation. And most of the time we observe that the sympathetic pathway is activated when we have some irritant in our mouth and we don't want to engulf it. So at that time sympathetic activity will try to stop the release of saliva so that it will not enter into our stomach, so we will have time to spit it out. So in that case the blood vessels will be constricted so that less amount of bicarbonate potential will enter. So in this way, sympathetic activity is basically contributing towards inhibition. However, sometimes a slight increase in salivation can also take place just for the sake of safety, you can say. There are several functions of saliva. I am just quickly have a look on these: facilitation, swallowing, and speech. If the saliva amount of saliva is less in our mouth, then what will happen? It will be very difficult for us to speak, and of course we cannot swallow the food because the bolus is basically is made under the contribution of the saliva. By the help of chewing and saliva, all these things, the bolus will be formed and then it will enter into the esophagus. So this it helps in the swallowing as well as speech. Cleaning, hygienic role is also very important of the saliva. It will clean the mouth and the teeth by washing, antibacterial effect by the help of the enzymes which are present, lysozymes and IgA. It can also function as the buffer. As I already told you, bicarbonate and phosphate are buffers that can make the saliva basic in nature. And then we can have the role of saliva in the digestion of basically the starch and the lipid. So the amylase which is present in the saliva is contributing towards the digestion of the starch, and the salivary lipase is basically lipase is for lipid digestion. Okay. So and then we have excretory function for the waste products like lead, mercury, iodide, fluoride, and some drugs and alcohol. So it will help in excretion of these products also. And then it will facilitate the taste sensation. Jo the main thing, the you can say, some people the purpose of the life is to eat, to taste the food, taste sensation. They spend all the money they are earning on the food. So Of course, the taste is very important, and the saliva is all contributing to the sense the taste of the food. Regulation of water balance. Sometimes decrease with the help of the saliva, decrease in dehydration and give thirst sensation. For example, if we have a dry mouth and if we are not able to sense it, then how can we balance the decreased amount of water? For that purpose, it will give the sensation. As a result, we will drink water. Basically, here you can see the repetition of these functions because I was in hurry. I think maintaining the well-being of the mouth, protection, of course, from the bacteria, buffering action, digestion, facilitation to the test, defensive action of the microbes, ionic exchange between tooth and the surface. Here, what does it mean? Ionic exchange between the tooth and surface is basically the calcium exchange that can make the teeth strong. Okay. ओरल हाइजीन के अंदर तो मेन कंट्रीब्यूशन है वन ऑफ दी saliva contains several factors that destroy the bacteria. One of these is the thiocyanate ion. It is very important ion that contributing by destroy the bacteria. Another is several proteolytic enzymes like the enzyme which can destroy or break down the enzymes and will break down the proteins. So proteolytic enzymes like lysozyme is present that attack the bacteria, right? Aid the thiocyanate ions in entering the bacteria, where these ions in turn become bactericidal and digest the food particles, thus helping further to remove the bacterial metabolic support. Saliva often contains significant amount of antibodies that can destroy oral bacteria. जैसे मैंने कहा था IgA मौजूद होगा, ठीक है, and some including some that cause the dental caries. So whatever types of bacteria is present in the mouth, saliva will produce antibodies, thiocyanate ions, lysozymes, all these contributing in the cleaning or the hygienic purpose of the mouth. In the absence of saliva, oral tissues often become ulcerated and otherwise infected, and caries of the teeth can be become revamped, rampant. So this is about our today's lecture. I hope you have understand everything. This is very simple and easy lecture. You just have to go through one time, I think, or two times maximum, and you are good to go. So this is my favorite picture in the whole lecture. How the isotonic fluid is becoming hypotonic, and how the movement of the ions are moving. So with this, thank you so much. Good luck. Allah Hafiz.